I'm Jackson and this is Bonnie, and we did our research project on the gut microbiota and behavioural issues in production, performance and companion animals, a systematic review.
Right, okay, for years humans have been using sayings like "we're scared shitless," swallowing our disappointment, or needing time to digest a defeat. We may also be left with a bad taste in our mouths after a nasty comment, or with butterflies in our stomachs if we're feeling a bit nervous. Now, despite humans having recognised this connection between our feelings and our gut for some time, science has only relatively recently begun exploring the influence that the gut has on our feelings as well as cognition and behaviour.
So of particular interest in our study was the gut microbiota. So this is the part of your gastrointestinal tract from your mouth down to your colon, and it includes trillions of little microorganisms such as fungi, viruses, single-celled organisms, and in our study, particularly bacteria. So we focus solely on the bacteria and their relationship and communication with the brain in what we call the gut-brain axis. So in a healthy animal, it's said to be in a state of homeostasis, where you have a balance between the good bacteria and the bad. But in the event of a stress, of infection, of changes in diet, and even the administration of antibiotics, can sort of mix up this balance that we've got between. With those that are favouring the change, they'll eventually end up proliferating. So this is known as gut dysbiosis. It can result in the disturbance in communication between the gut and the brain, and that's increasingly recognised in disorders such as Parkinson's, autism, obesity, and other mental health disorders. For our research, we focus specifically on mental health conditions, including depression, anxiety, bipolar disorder, and schizophrenia. While these disorders aren't recognised in animals, we do know that there are a lot of behavioural issues that animals can undergo, and we believe that the gut microbiota is associated with that.
So the purpose of our research was to analyse the current gut microbiota research that's available in animal studies, and essentially provide the first correlation of this by comparing three categories of animals. So we looked at production animals with pigs, performance animals with horses, and companion animals with dogs. Now, at the same time, we also looked at the most recent human literature to understand the putative mechanisms of dysregulation. And also we wanted to compare our findings of the animals with those already identified in mental health patients in humans. And finally, we're also hoping to assess whether there may be an opportunity in the future to target the gut microbiota as a way of improving some of these abnormal behaviours in animals. And so for our systematic review, we created search terms like this one for each of the species, and we used this in PubMed to identify relevant articles. And if you look at these graphs, you can see that over the last 20 years, there really has been an explosion of interest in this area in human research, with over 10,000 results generated. If you compare that to the animal research, where we had a combined total of about 300 articles, I guess this really highlights that it is an area of research that's really in its infancy. And so after sifting through these articles, we identified 39 that we chose to look in depth and include as part of our systematic review.
So, the gut bacteria can help us in many ways. They break down our dietary carbohydrates into short-chain fatty acids, they help our brain cells to develop, they help build and maintain our immune system, and they produce many of their own products which can travel through the bloodstream to different places, causing different effects. We as people and as animals can also have an effect on our gut bacteria, with our stresses, our diets, and our antibiotics influencing both the population and the effects that the bacteria have. This two-directional pathway is called the gut-brain axis. Some of these short-chain fatty acids, the neurotransmitters, and the precursors of these molecules will have an impact on our brain function and our central nervous system, which can then impact our emotion, our moods, and our behaviour. There are specific bacteria within the phylum Bacteroidetes and Firmicutes, both of which will feature in many of our behavioural issues in animals, which will ferment some of our food down into short-chain fatty acids such as acetate, butyrate, and propionate. Another key product to look out for. So these are used for a massive list of things, but we're focusing mainly on their effect on the brain and the central nervous system. They can cross the barrier between the blood and the brain, causing an anti-inflammatory and a pro-building effect on specific actions of the brain that are involved in cognition, in emotion, and in behaviour. Butyrate specifically is a crucial short-chain fatty acid which keeps our gut lining intact and acts as a precursor of many microbial metabolites. Neurotransmitters as well, such as dopamine and serotonin, are widely known to influence our emotions. But the same can actually be said of animals. Dopamine is known as the pleasure hormone and is associated with motivation and reward, while serotonin is known as our happiness hormone, which is associated with mood and sleep. And GABA is associated with having a calming effect. While it certainly seems like these tiny bacteria hold a lot of power over us, in a healthy individual, they're definitely our friends. It's only when we disrupt their habitat that things tend to go a little bit sideways. When we affect the microbiome, we can either cause the destruction of good bacteria or the proliferation of the bad bacteria, or a combination of both.
And so within the human articles, we looked at depression, anxiety, bipolar disorder, and schizophrenia. And across these articles, we noticed that there wasn't necessarily a significant difference in the richness of specific bacteria within these patients, which we term alpha diversity. But there was a difference in beta diversity, and so this is represented as there was a significant reduction in the overall biodiversity in mental health patients when you compared them to controls. And when we had a little bit of a closer look, we identified that firmicutes, the phylum Firmicutes, represented the most frequently reported change amongst the articles. But interesting to note that there was a relatively even spread between those reporting increases and decreases. And so when we took a closer look within this phylum, we actually noticed a few trends, and that was those that were contributing to the increases were often lactic acid producing bacteria. And so when these proliferate, lactic acid accumulates in the gut, which has been known to change the environment to one that benefits pathogenic or bad bacteria. We also noticed an increase in GABA-producing bacteria. And like Jackson said, GABA is known as the calming neurotransmitter, but it's actually been found when it's at really high concentrations, it can actually have effects associated with anxiety and depression. And finally, we noticed associated with the decreases seen in this phylum were often a decrease in butyrate-producing bacteria. And like Jackson said, butyrate is one of those essential short-chain fatty acids and has a key role in maintaining the gut barrier. So essentially when this decreases, bacteria as well as other things in the gut can more easily escape and enter the bloodstream, where they have the potential to cause systemic inflammation.
So now we're going to look at our animal models. Poor nutrition is commonly known as a major causation of behavioural issues, but it's only really in the last 2 to 3 years that the science has dived more in depth in the microbiota in the gut. So with poor nutrition specifically contributing to the microbiota rather than just the food causing the change. So in our review, pigs were chosen as the representative for our production animal industry. Tail biting specifically is a widespread behavioural issue among the industry, with pigs often responding to stress, to boredom or to frustration by biting each other's tails. A recent study estimated that it costs around about $23 per pig per year in the industry, which causes a loss of millions of dollars over the whole industry. We found the main effector of the shift in gut bacteria was the Firmicutes phylum, which we discovered was a common pattern among all of our animal groups. We found that pigs and humans had similar results, with the relative increase and decrease being approximately the same, showing a relatively even split. So one species that I would like to highlight is actually the Coprococcus species. This is found in pigs that are displaying stress and anxiety behaviours. But the specific bacteria Coprococcus is known to coincide with genes that produce dopamine, which is known as the pleasure chemical. So we found that pigs, opposite to humans and mice, had an increase in Coprococcus, whereas humans and mice had a decrease. Therein lies one of the most difficult yet exciting aspects of this literature review. We found correlations between the bacteria and the behavioural changes, but we're only really on the cusp of their true meaning.
Okay, and so horses represented our performance animals, and they're really a prime species that examples where modern day management has really differed from their traditional way of living. Nowadays, horses, instead of spending a lot of time on pasture, competition and performance horses often receive large amounts of grain. And often in these horses you can see abnormal behaviours like hyper-reactive horses, as well as an increase in some of the stereotypical behaviours like pacing and windsucking. When we looked at the gut microbiota of some of these horses, we've noticed similar changes, and that was that Firmicutes again were the most frequently reported change. But unlike pigs and humans, this was predominantly reported as an increase. One such being an increase in Streptococcus, which was found to be consistently abundant in alert horses. Streptococcus is one of those bacteria that's known to produce lactic acid, and interestingly it's also been associated with laminitis in horses. Now, Streptococcus is also known to produce serotonin, which is known as the happiness hormone. But actually, when it's in really high concentrations, serotonin's been known to be associated with restlessness, agitation, and even increased reflexes in mice models and in humans. So we hypothesize that this may present itself as this hyper-alert behaviour in horses. And finally, for companion animals, we looked at dogs. And similarly, they also had lifestyle changes, including diet was mostly linked with increases in some of these behaviours we're seeing, including aggression and phobia. And so, like a lot of the other species, there was again a reduction in the biodiversity in these dogs showing abnormal behaviours. And again, increases were most, changes in the firmicutes were most frequently reported. And similar to horses, this is reported as an increase. And when we took a closer look, we noticed similarities with humans with mental health disorders in that the increases were mostly attributed to increases in lactic acid producers and GABA producers. One example of this is the genus Lactobacillus, which was found to be consistently overabundant in both phobic and aggressive dogs.
So, now we've got a very complex phylogenetic tree here, which demonstrates the specific examples of where anxiety and depression and other behavioural orders in humans will also coincide with ones in animals. So we've got some specific examples in the phylum of firmicutes, which is tail-bitten pigs were sharing exclusive change with humans with depression, having a relative increase in the bacterial species of Blauscher. We also found that phobic dogs share similar increases in lactobacillus as individuals with depression and anxiety. And withdrawn horses will show other elevations, which is a change solely shared with humans with depression.
And we also found similar findings like this in other phyla where specific microbiota changes were exclusively shared across species displaying these depressive-like behaviours. And so this really demonstrates that there's not only an association that exists between microbiota changes and behaviour in individual species, but that we can also apply this across species when we're comparing similar behaviour.
While there are a plethora of factors that can change and affect the gut microbiome, there are some preventative measures that can be taken. However, the research is still in its very early stages, and for the sake of time, we're unable to talk about this in our presentation, but feel free to question us afterwards.
Okay, so to summarise our findings, we were able to demonstrate that there was an association between the gut microbiota and abnormal behaviours within and between our production, performance and companion animals. And as we've seen in humans with mental health disorders, animals exhibiting these abnormal behaviours were often found to have an increase in the lactic acid producing bacteria, as well as a decrease in the butyrate producing bacteria. With our current research, however, we have a situation a little bit like the chicken and the egg scenario where we're actually unable to distinguish between cause and effect. And so our findings really indicate three possibilities. One, that abnormal behaviours result in physiological changes that alter the gastrointestinal environment and influence this dysbiosis. Or two, specific microbiota may influence the development and the severity of some of these abnormal behaviours. Or three, animals with abnormal behaviours are predisposed to a confounding factor that is also impacting the microbiota.
So this left us with a foundational paper gathering evidence of the current snapshot of the scientific community. And we hope that our paper can be used as a platform for the development of other novel therapeutic and preventative interventions. We do have a gut feeling, however, that the gut-brain axis will be understood to a much greater extent in the future, much to the health and benefit of all animals. Thank you.
Thank you, Bonnie and Jackson. Whoa, we've boom. We've got Beck first, Kapil second, and then we'll see how we go with Ryan and Eduardo. But Beck, go for it.
Great talk, Ryan. It was really good. Very informative. I just wanted to ask that in your research, did you come across any papers that looked at maternal seeding and how these types of conditions and a change in biodiversity microbiome actually set up during that in utero or in ovo development, and then maybe the size environment, there could be a genetic link between even grandparents, parents, and their progeny in terms of displaying these types of behaviours?
Absolutely. I think the majority of research, I know for me specifically in pigs, was looking at the influence of parturition and the effects of caesarean sections versus natural birth and how that affects the gut microbiota. So we did find, and we spoke about it in our manuscript quite at length, that, yeah, parturition will directly affect the type of gut bacteria that will be in, for example, piglets' guts. And there are obviously big changes as you go through life. There are 3 distinct stages of life where the gut bacteria will change. But yeah, absolutely, genetics does play a big role in it.
And in the human research, it seemed to be that's the real recent studies have been really emphasising the gut microbiota disturbance in utero, and actually suggesting that they actually have permanent effects on behaviour if it happens in utero.
Yep.
Yeah.
Just notice we're a little bit over on our time, so we've got 3 very interesting questions. Can you guys check with these guys
Yeah, come see us,
afterwards?
yeah.
Yes.
Yeah.
So just in fairness to other speakers, but Kapil